Principal Ma'am, below friends. I welcome you all cordially for the workshop and on mushroom cultivation.
Okay, mushroom etc. But you know well, as a biotechnology student, you know that mushroom has lot of nutrients, right? Especially protein, like protein, highly protein content. I appreciate the department head as well as all the faculty members for initiating this wonderful program, especially to initiate the startup. It's nothing but like a startup. Suppose at the end of the day, there is a time span of cultivating mushroom. It will take 1 month to complete the cultivation. And I advise all of you to make use of the areas which are ideal, not used for any purpose. So you can make use of that area, you can choose, and with the guidance and support of your HOD ma'am, you can jointly cultivate the mushroom as a group event. And that area will be promoted as startup of incubation center of your department as well as the college. Okay, that incubation center என்ன தெரியுங்களா, ஸ்டார்ட் அப்க்கு முன்னாடி, அதாவது ஒரு பிசினஸ் பண்றதுக்கான ஒரு ஐடியாஸை கொடுத்து உங்களுக்கு பிசினஸ் ஸ்டார்ட் பண்ணணும்னு வச்சுக்கோங்களேன், அதுக்கான செட்டப்போடு இருக்கக்கூடிய ஒரு ஏரியாவை தான் நம்ம என்ன சொல்றோம், இன்குபேஷன் சென்டர். I appreciate and congratulate Mahaalakshmi ma'am for initiating this program, because it will be included in incubation center. Like, so when we go for accreditation like NAC, we can show this mushroom cultivation as incubation for our institute by our students. So one day I hope day by day you will be learning to grow this mushroom, cultivate this mushroom plant in a better way, in a mass area in our college. And I advise all of you to make use of the area. Thank you very much. I appreciate you all also. Thank you.
Thank you, ma'am. Now I request Vice Principal ma'am to facilitate the gathering.
First of all, I congratulations to Head of the Department of Biotechnology and all the faculty members of Biotechnology Department and my students also. Mushroom cultivation is very famous nowadays. I think mushroom biryani is very famous latest. Okay. நிறைய பார்க்கறதுனால நான் சாப்பிட்டதில்லை, ஆனால் யூடியூப் சேனல்ஸில் நிறைய வருது. அது மஷ்ரூம் பிரியாணி ஃபேமஸ் தான். அதுக்கப்புறம் கேரட் நான் அட்டன் பண்ணேன். கொஞ்சம் எனக்கு தெரிஞ்ச வரைக்கும் அந்த மஷ்ரூம் வந்து கொஞ்சம் ஃப்ரெஷ்ஷாக 1 or 2 days அதை நம்ம யூஸ் பண்ணணும்னு சொல்கிறாங்க. கொஞ்சம் பிளாக் கலரோ இல்லை ரெடிஷ் கலர் கரெக்டாக கொஞ்சம் ப்ரௌன் கலர் அந்த ஷேட் ஆகிருக்கா கூட அதை யூஸ் பண்ணக்கூடாது, பாய்ஸனாக மாறிடும்னு சொல்கிறாங்க. கரெக்டுங்களா? எஸ். அதனால வந்து ரொம்ப கேரக்டாக அதை வந்து யூஸ் பண்ணணும். சரிங்களா? So not only we are studying in the classroom teaching, so class just we are teaching the theoretical knowledge only like theoretical knowledge better than but in the workshops organized for both matter we have practical. Okay, practical ability mushroom kind 1 month or not time spend பண்ணிப்பாங்க நினைக்கிறேன். 1 month or 1 week, 1 month or 1 week எப்படி பண்ணணும்னு காமிக் பண்ணுவாங்க. ஸோ அது மாதிரி அவங்களோட ப்ராக்டிக்கலாக நீங்கள் இருந்தீங்கன்னா நீங்கள் கூட மேம் வந்து பண்ணும்போது ப்ராக்டிஸ் பண்ணும்போது பார்த்தீங்களா எப்படி அதெல்லாம் சீட்ஸ் போடுறாங்க என்ன எதுன்னு பார்த்தீங்களா இருக்கும் அதை எப்படி தெரிஞ்சுக்கோங்க மார்க்கில். சரிங்களா? எப்படி அதை கல்டிவேட் பண்ணாங்க என்னன்னு சொல்லிட்டு உங்கள் ஸ்டாப் நீங்கள் தெரிஞ்சுக்கோங்க. அந்த மாதிரி நீங்கள் ஒரு ப்ராப்பராக ப்ராக்டிக்கல் நாலேஜ் கைட் பண்ணும்போது நம்ம ஒரு ரீசெண்டாகவும் நம்ம என்ன பண்ணிக்கலாம்னு பார்த்தீங்கன்னா யூ ஹாவ் டு டெவலப் செப்பரேட் பிஸ்னஸ். இண்டிவிஜுவல் நீங்கள் பிஸ்னஸ் பண்ணுறதுக்கு இது ஒரு வாய்ப்பாக இருக்கும். அப்படின்னா ஸோ ஐ கேன் டு பயோடெக்.
Our request Principal ma'am and Vice Principal ma'am to visit the mushroom grow.
let's see the practical session of how to prepare the mushroom bed. The sum of the precaution to be taken during mushroom cultivation are: strict hygiene should be maintained inside the farm. Never use the broomsticks for cleaning dust inside the farm. Keep an eye on temperature and humidity percentage inside the room, because the temperature and humidity is very important to cultivate the mushroom. Use only the filtered air inside the peak heating, spawn running and cropping rooms. There are many importance in mushroom cultivation. Mushroom cultivation is so important as it reduces vulnerability to poverty and strengthens livelihoods through the generation of a fast yielding. It is a nutritious source of food and reliable source of income. Never lose hope. Mushroom cultivation may take time to perfection, but there is no point as a loss here. It does not require access to land. Its cultivation is viable and attractive for both rural and urban farmers. The cultivation of oyster mushroom is usually carried out in a transparent polythene covers. The size of the cover should be 60 into 30 cm with a thickness of 80 gauge. Wash hands thoroughly with the antiseptic lotion. Then take the polythene cover and tie the bottom end with a rubber band and turn it inwards. Soak the paddy straw in cold water for about 4 to 5 hours and shade dry steam sterilized paddy straw to get a uniform moisture level in all areas. Take out a well-grown bed spawn, squeeze thoroughly and divide it into two halves. We can prepare two beds from the single spawn bag. Fill the straw to a height of 3 inches in the bottom of polythene bag. Take a handful of spawn and sprinkle over the straw layer, concentrating more on the edges. Fill the second layer of the paddy straw to a height of 5 inches and spawn it as above we have done. Repeat the process to get 6 to 7 straw layer with the spawns. Gently press the bed and tie it tightly with the help of rubber band. Put 6 ventilation holes randomly for ventilation as well as to remove the excess moisture present inside the bed. Plug the sterilized cottons in the holes to avoid the entry of insects. Now finally arrange the beds in the thatched shed following hanging rope system. The temperature should be maintained at 22 to 25 degrees Celsius and relative humidity level is 80 to 90% inside the room. Observe the beds daily for contamination if any. The contaminated bats should be removed and destroyed. Similarly, observe regularly for the infestation of insect, pests, flies, beetles, mites, etc. Then the spawn fully runs on the bed and bud will start to produce. Then spray the water to keep the bed in moist level. After bud formation, the mushroom will grow and we can harvest it at a certain days. Now let's see about the diseases caused to the mushroom bed.
Mushroom disease can be divided into four types: black mold disease, orange mold disease, green mold disease, and yellow mold disease. Now I am going to explain about first black mold disease. Black mold disease, also known as pinhead mold. Black mold is a common mushroom contaminant that is common in soil and air, and it is one of the first molds to appear on stale bread. This fast growing fungi has a dense mycelium that is white at first and becomes grey, then takes on a black appearance. I am going to explain about green mold disease. Green mold disease scientific name is Trichoderma. Trichoderma is a group of green mold fungi that is present in all soils, with several species having the ability to form symbiotic relationships with plants and plant roots. One species in particular, Trichoderma herrerianum, is one of the most common forms of contamination seen in mushroom cultivation. Next, I am going to explain about orange mold disease. Orange mold or red mold or pink mold are alternative names for Neurospora grassi, another common and extremely fast growing fungal contaminant. While used for decades in genomics research, this bright neon orange mold can wreak havoc on your operation in as little as 8 to 12 hours. It first appears as an orange-white wisp but quickly develops into a bright orange powdery patch. If allowed to grow, this patches produce round lumpy formations, but it is best to catch the fungus prior to the stage. Once found, contain the source by sealing it in a plastic bag and disposing of it immediately.
Very good morning to everyone. Today I am going to explain about the mushroom. First I am going to introduce to you what is a mushroom. Then I am going to share information about its structure, life cycle and its types. Yes, everyone likes to eat mushroom and most of the vegetarians people eat mushroom to get the equal protein level. But we don't know about how mushroom grows and its additional benefits. Mushroom is a fungi, fleshy fruiting body arising from the group of mycelia buried in a organic matter. It is not a plant, it is uniquely different from the plants. It consists of the two parts. The cap-like structure is known as phylus and the thread-like structure known as mycelia. Mushroom does not require sunlight. It is a non-photosynthetic. It grows under rocks and in mountain region, tree trunks, soil rich in organic matter. The spores of the mushroom is made up of the chitin, which is the hardest naturally made substances on earth. Its classification. Mushroom belongs to the kingdom fungi. It belongs to the genus Pleurotus, species Ostreatus. Phylum is a Basidomycetes. Class is a Hymenomycetes, order is a Agaricales, and family is a Tricholomatesia. Now I am going to tell about its structure. Mushroom is composed of a hair-like cells that make up mycelium bunching together to develop into primordia or a baby mushroom. The first part is the mushroom spores. Mushroom spores are called basidiospores. The microscopic seeds act as a reproductive agents. It fall on the substrate to produce the new mushroom. Spores are very small and mostly colorless. When the spores gather into cluster, they will display similar color to that of the gills. The next part is a stem. It is a axis supporting the mushroom's cap. The stem of each type of mushroom vary in size and length. Usually stems are cylindrical. The bottom part of the stalk is big and gets slimmer when it nears the top. Top part of the stem joins the cap or gills. The skin outside the stem of some mushroom is rough, while some are scaled like nets. Inside the stalk, some mushrooms are loosely woven up with fiber similar to the sponges. The next part is the gills. The underside of the mushroom cap that has a thin sheets connecting in a row around the stalk and extend upward to the tip of the mushroom cap. The gills are connected to the stalk. The next part is the cap. The top part of the mushroom that grow upwards above the stem is the cap. When it fully grown, the mushroom cap will spread out like an umbrella. Most of the growth of the fungi does not noticed because it occurs in underground. Mushroom actually takes days or a weeks for one to develop. Now next I'm going to explain about how the mushroom grows. The life cycle of mushroom has four stages. The first stage is spore germination, second is colonization, third is forming fruit body, and fourth is spores sporulation. First, mushroom produce and release spores from the gills that are located underneath the mushroom cap. Mushroom produce hundreds or thousands of spores. Think each of the spore as a single cell, and this spores is either male or a female that ultimately group up with other spore to create a male and female hypha. Hypha of one spore meet with the hypha of another spore and produce the mycelium. Mycelium is a root-like structure, provides a stable and nutritious area for the fungi to start producing mushroom. Upper surface of the mycelium, where small fruit body is formed, is known as the primordium formation, and the mature fruit body will form. Now I am going to explain about the types of mushroom. There are various types of mushroom such as oyster mushroom, Agaricus bisporus, Flammulina filiformis, penny bun, hen of the word, Clanterella mushroom, Agaricus campestris. In India, we mostly cultivate oyster, button and milky mushroom. First, I am going to explain about oyster mushroom. The scientific name of a oyster mushroom is a pleurotus. Oyster mushroom is a good source of the dietary protein. It converts the lignocellulosic waste materials such as rice waste, paddy straw, coconut fiber, bagasse waste, which is a byproduct of sugarcane industry, corn waste, into the dietary food material. Oyster mushroom was first cultivated in 1917 at Germany. It can be grown at a temperature of 20 degrees Celsius to 25 degrees Celsius. It requires humidity level from 80 to 90 percentage. It can also grow in the temperate and the tropic regions, which is not extreme cold and not extreme hot. It can grow in dead and dying wood. This mushroom is loaded with the fiber, vitamins, minerals, and other important nutrients. They contain low carbohydrate dietary protein. Oyster mushroom reduce heart disease by lowering the cholesterol and blood pressure. Mushroom decrease the pathogenic bacteria and increase the production of short chain fatty acid in the gut. Its classifications are Its scientific name of oyster mushroom is Pleurotus species. It belongs to the family Polyperaceae. Genus is a Pleurotus and species is Sager Sapidus ostreotus Florida flabellus. Its class is a Basidiomycetes and belongs to the subclass Holobasidiomycetida. Now I am going to explain about the white button mushroom. The scientific name of the white button mushroom is Agaricus bisporus. It is a thumbnail sized and it is like a button shaped mushroom. We can eat it by both raw and cooked. When it is raw, its flavor is mild, that is no smell or no taste, but after it is cooked, it has more fragrant and tastes like a meat. It is either white in a color or brown in a color. It is free of calorie. It contains nutritional substances such as potassium, selenium, and proteins. It also contains cancer preventing antioxidant. They grow in cold regions. Temperature for forming the fruit body is about 14 degrees Celsius to 18 Celsius. Humidity level is 80 to 90 percentage. This mushroom is commonly found in field and grassy area following rain, spring to autumn season. Its classification are: this mushroom belongs to the kingdom fungi. It belongs to the genus Agaricus and species Bisporus. Phylum is a Basidiomycota. Class is a Agaricomycetes. Order is the Agaricales and belongs to the family Agaricaceae. Its applications are, as I already said, we can eat it by both fresh or cooked. Their small size makes them ideal for skewer. We can serve it in a soups, stews, or salad. They can flavor with the other ingredients as well. It pairs up with the cheese, herbs, meats, wines, cream based sauces. Now I am going to explain about the milky mushroom. The scientific name of the milky mushroom is Calocyba indica. It is a robust which is a healthy, fleshy, milky white in color and it's shaped like an umbrella. It is suitable for both hot and humid climate. It grows well in the 25 to 35 degrees Celsius. It can be cultivated throughout the year. Its cultivation is very simple and easy. It requires low cost. The harvested period after spawning is about 24 to 28 days. The crop cycle from the starting to the end is about 45 to 50 days and its shelf life after harvesting is about 3 to 5 days. Its classifications are: it belongs to the kingdom fungi and genus is Calocyba. Division is a Basidiomycota. Class is Agaricomycetes. Order is Agaricales and family is Liphalaceae.
But we don't know how to differentiate it into edible or poisonous mushroom. Now I am going to explain how we can differentiate the edible and poisonous mushroom. First, let us go through some edible mushroom. Edible mushroom do not contain poisonous effect on human and it contain desirable taste and good aroma. Edible mushrooms are consumed by humans for their nutritional value such as carbohydrate, potassium, protein, dietary fiber and vitamins are present. Fat, cholesterol, sodium are absent in edible mushroom and they occasionally consumed for the supposed medicinal value. Edible mushrooms are fungal species that are harvested wild or cultivated. For example, Agaricus bisporus, Agaricus bitorquus, Pleurotus florida, Calocybe indica, Valvarella valvulaceae. In this, Agaricus bisporus is the common white button mushroom because morphology look like a small button. It is first cultivated in USA in 19th century. And then the next one is Pleurotus florida is the another common edible oyster type mushroom. It is first cultivated in 1970 in Germany. It is act as a medicinal mushroom because the shape of the spore cork represent oyster. And then the last one is Valvarella valvulaceae is a type of padistra mushroom. It is important variety that is cultivated throughout India. It is first cultivated in Coimbatore in 1943. Now let us go through some poisonous mushroom. In the morphology and life cycle, it look like a edible mushroom. They are all distinguished by following features. The fruit body is brightly colored. It contains greenish teenage and the gills are yellow-green color. Pink colored spores are present in gills. A presence of vulva and annals on the stalk. It contains unpleasant odor. The most common poisonous mushroom is Amanita phalloides. Is an important poisonous mushroom. It is deadly poisonous as a toxin which is known as amanitin. Consumption of this amanitin phalloide can cause fatality, means it causes death of a person. Other important poisonous mushrooms are fly agaricus, deadly dapperling, destroying angles. So we have to ensure before consumption the consuming mushroom is edible or poisonous. So let us go through some key steps involved in mushroom production. The mushroom production takes place between 1 to 3 months. We need to identify a clean and separate room for mushroom cultivation. In that room, the temperature, moisture, and sanitary conditions are maintained. We need to store the raw materials used for mushroom cultivation in clean place. And we need to pasteurizing. Pasteurizing means sterilizing the medium, bags, seeds, and spawns before the use. We need to water the substrate to maintain temperature and moisture condition. And then harvest and heat, the processing and packing, and the mushrooms are sold.
Now, let us see about the overall flowchart involved in mushroom cultivation. First, we need to choose the padistra as raw material because padistra contain high cellulose content. The cellulose is helpful for providing nutrient in mushroom. Then chop the padistra into 3 to 5 centimeter. Fill the chopped padistra in gunny bag and soak it in water for 8 to 12 hours. Drain the excess of water. Then treat the padistra with hot water and drain of excess water. After we want to dry the padistra for 60 to 70% moisture. The padistra should contain 30% of moisture content. Now the next step is to prepare the bed by adding padistra and spawns. Then pin the bed with the help of sterile needle. After 23 days of spawning, pinned is appeared, and after 28 days, the mature mushroom are formed. Then the harvesting is done. Now it is time to pack the mushroom and sell it in the market. The mushrooms are sold in market both in fresh as well as in dry.
And then finally, we see about the importance of mushroom. First, we see about the nutritional value involved in mushrooms. Mushroom contain protein, vitamin, and minerals. Is beneficial to person health. Protein is high in vegetable, fruits, and pulses. It contains vegetable protein like threonine, valine, methionine, tryptophan, leucine, and isoleucine. It is also act as a antioxidant. Mushroom contain vitamins. The vitamin D is present. It is helpful for bone health, immune function, weight loss, and prevent heart disease. Vitamin B2 is also present. It is helpful for to maintain healthy skin. Vitamin B3 make hormones. Folic acid also present in mushroom. It helps for production of blood. Potassium helps for healthy blood pressure and muscle and nerve activity. And then finally, phosphorus also present in mushroom. It helps for healthy bones and teeth. The mushroom is cholesterol free, sodium free, and calorie free.
Now we shall compare the nutritional value of mushroom with meat.
The nutritional value present in meat is low when compared to mushroom because mushroom contain all the nutrients like protein, carbohydrate, sugar, fiber, calories, but meat only contain protein. Next we see about the medicinal value present in mushroom. Mushrooms are probiotic in nature. Mushrooms are used as a medicine in the pandemic disease. It has a medicinal value like it helps in low calorie and it contains high vegetable protein, fiber, iron, zinc, vitamin and minerals. It produces healthy human body. It helps to lower blood pressure. In combination, they strengthen the immune system and aid in neuron transmission, metabolism, hormonal balance, and the transport of nutrient and oxygen. And it possesses anti-allergic, prevent allergies, prevent the formation of tumor cells and avoid cancer, that is anti-tumor, and it is also act as a anti-cholesterol. It is also used to treat skin disease. And finally, we discuss about economic value. Mushroom cultivation is easy with shorter crop cycle, and mushroom growing has got high employment potential. About 80% of workforce engaged in mushroom is women. Mushroom cultivation is good enterprise for small farmer and landless farmer. Our country can earn foreign exchange through export of fresh mushroom. And then the other uses are mushroom is used as biofertilizer. After the harvest of mushroom, the beds are used for biofertilizer making. It is also used as a dyeing hole. Before the invention of synthetic dye, mushrooms used as a textile dyes. It is also used as a natural fiber, and then it is also used as fire starter. Types of polypores, example tinder fungi is used as fire starter. Mushrooms is used in biological remediation technique by lower the level of contaminated water. These are all the mushroom and its importance.
Hi everyone, in this video I am going to explain about the process of mushroom seeding. It follows two methods. One is a pure culture preparation. Materials required for the pure culture preparations are potatoes, dextrose, agar, measuring cylinder, test tubes, conical flask, gentamicin, cotton, ethanol, forceps, and sample of oyster mushroom. Measure the 500 ml of distilled water using measuring cylinder. Boil the 200 gram of peeled potato in 500 ml of distilled water for 30 minutes. Twist and roll the cotton in test tube. Plug the cotton tightly to prevent the entry of microorganisms. Test tubes are kept in a beaker and wrap it with a newspaper and kept it in the pressure cooker for about 15 to 20 minutes for the purpose of sterilization. After 30 minutes, we can see the potatoes get boiled. Then let the potatoes get cooled under normal temperature. Now after cooling out, we just have to filter out the potato extract with the help of filtrate or a clean cloth. Measure 10 gram of dextrose and 10 gram of agar in a weighing machine. Add it in a potato extract. Keep it in a pressure cooker for 20 minutes for proper dissolving. Now the PDA medium is prepared. Place the medium in laminar airflow. Add 3 drops of an antibiotic named gentamicin. This antibiotic was added to prevent the contamination of microbes. Hold the conical flask with the lid pointing towards the test tube. After, pour the media in the test tube. The mouth with the cotton plug. After pouring the media, the test tubes are kept in a pressure cooker for about 4 whistles at 20 minutes. This again and again sterilization was done to prevent the entry of microbes. After sterilization, the test tubes are kept in a slant position in the laminar airflow. This slanting the surface of media is allowed to solidify. After solidification, keep the test tube in inverted position for 2 hours. Especially the lower portion of the cap where the gill plate joins the stem is considered the best tissue for excision. The mushroom is cut lengthwise from cap downwards with a flamed needle. A small piece of the internal tissue of the broken mushroom is cut out The cutout tissue is inserted into the culture media test tube with the help of forceps. The inoculated test tubes are incubated at 25 to 30 degrees Celsius, preferably in darkness for about 7 days. Now the next step is spawn production. Materials required: forceps, cotton, calcium carbonate, sorghum seeds, polypropylene cover. Wash the sorghum grains and cook the grains in pressure cooker without putting vessel for 30 minutes just to soften them. Take out the cooked grains and spread evenly over a hessian cloth to remove the excess water. Mix 20 gram of calcium carbonate thoroughly with the cooked dried grains. Fill the grains in polypropylene bags up to 3/4 height, approximately 300 to 330 gram per bag. After filling the grain, a polypropylene neck is placed near top by passing PVC ring and folding back the edges of the bag down. Plug the mouth tightly with non-absorbent cotton wool. Cover the cotton plug with a piece of a paper and cover it tightly around the neck with the rubber band. Arrange the bags inside an autoclave or pressure cooker and sterilize under the pressure cooker for about 20 minutes. After sterilization, the seed package are taken into laminar airflow. Then the mother tissue culture from the test tube is transferred into 1/2 portion of a bag with the help of forceps. Similarly, transfer the mother spawn obtained from pure culture into the another bags for commercial spawn production with the help of forceps. Plug the mouth of the seed bag with the cotton plug. Incubate the inoculated bags in a clean room under room temperature for 10 days for the further use to prepare the bed spawn.
I hope you are all understood. Thank you.